5, 4, 3,
What
2,
do you mean I can't eat cookies
1.
anymore?
You can eat the cookies. Chew on the cookies. I see you all chewing on the cookies.
I'm
How is
having
it
dark
sounding?
chocolate with sea salt. So
How is this sounding? That's delicious. Yo, yo, yo. Are we popping? Are we clicking and popping? Did I change us? For
oh,
no
we're having
reason?
trouble streaming to Twitch.
What? Are we?
Yeah, I just got a pop-up from StreamYard.
Why? I didn't. Why did
This
you
may
get
be an
it?
issue on Twitch's end. We'll keep trying and let you know if it's resolved. There's not a little exclamation point next to Twitch in the upper right?
Nope. Everything is good. It could be the last
I wonder why
stream.
it says that then.
How weird. Yeah. Everybody says 5 by 5. Doing it. Oh, here. Now I finally got it. The Twitch. The Twitch you. Twitch you. Where did Justin go?
Oh, I'm still here. I was just washing down my cookies.
Cookies for later. Nothing greater.
Oh, cookie
than
now.
cookies for later.
Cookie starts with C.
Looks like it might be streaming. What is the problem there?
It looks like we're good. Let's rock and roll.
Yeah.
Take 2. Where were we?
You were talking about cancer.
Just good news, everybody. Cancer. So,
sorry, Rachel.
This is okay. I'll recap really quickly the story that nobody could hear. In the last 30 years, cancer mortality has fallen in half. At the same time, mortality remained unchanged at 5.9 deaths per 100,000 in the last 30 years because while mortality has fallen in half, it's largely because detection of new cancer has doubled. So incidents of cancer being recorded have doubled. Actual mortality per 100,000 has not moved.
So the same number of people are dying relative to population,
right?
But cancer is less deadly. So how many people did they did? It's less deadly in that, like, the number of cases relative to the number of deaths is different, right? But how many people were diagnosed with cancer via autopsy, right? Like, how many people were diagnosed with cancer when they were already dead?
Oh no, that was probably, that's a great point. That was probably much more.
They cut open and saw tumors everywhere, right? Like that's part of the detection.
So I'm still trying to figure out. So in this study, they have suggested that the higher incidence of cancer rates that we've been seeing through diagnoses are because diagnoses are way up. That we're detecting more types of cancer earlier, and that it has, it's not that there's a new cancer epidemic hitting, that it's the same cancers we've always had, same basically rates, I guess. It's just that our ability to detect them and detect them much earlier has improved. Okay. But then I have to question if it's what is it, 5.9, did I say? Deaths per 100,000 is 5.9 deaths per 100,000 is unchanged between 1992 and 2022. That tells me we've gotten no better at fighting cancer.
Interesting.
There are more cases. We're better at finding it and seeing it.
There's double the cases because of diagnostics.
But if there are double the cases, we're also better at treating it because we are keeping up with and continuing the decrease in.
You would then
deaths,
think that the deaths per 100,000 would go down. Now, the thing that Blair said, how many of these are via autopsy, is interesting because that throws in the possibility that We just missed causes of death previously.
Sure.
It's still, I mean, then you could say like, yes, actually the real number from 1992 should have been more like 10 per 100,000, but because we weren't so as good at diagnosing, we missed a lot of things that were cancer that killed people. But the number's unchanged, which is
there's another possibility.
Coincident.
It would be very coincidental, but it is possible that people are getting cancer more because of chemicals and PFAS and
Right.
whatever. Just name random things.
For sure.
High fat diets.
But that we are better at treating it. And the rate of increased cancer and the rate of treatment is like canceling each other out.
Exactly. No, we have to be better at treating it to be able to keep up with the wave of new diagnoses.
This current study, which is out of Harvard Medical School, Brigham and Women's Hospital, and Dell Medical School researchers are saying no, the increased rates of cancer, the doubling of the rates of cancer in the last 30 years is wholly due to improved screening and earlier detection.
Which is amazing, because even though the same number of people are dying, people are probably in less pain.
So there's another study out this week that is suggesting that the increase in cancer rates, colon cancer specifically, in younger adults is specifically related to diet. And they're trying to tie it to highly processed foods, which could be possible. There could be things, but we have this one study that came out and saying a lot more younger people are getting cancer. But what you're saying,
and it actually points out
diagnosing it more.
It actually here it states that colorectal mortality rose slightly, while its incidence rose much faster. So not everything moved together. Thyroid and kidney cancers display sharp incidence increases alongside stable or declining mortality patterns. So that's more consistent with potential overdiagnosis, finding lots of what you would call non-fatal or maybe non-dangerous cancers.
Mm-hmm.
Cancer happens all the time. It's not all, not all it gets to the point where it's actually life-threatening. It's a lot of things, sort of slides moving in different ways. The part that still is bothering me is that if it was like, it was 5.9 and now it's 5.5, if there was any difference in that mortality rate, I'd be like, okay, yeah, it's a bunch of different things at play. But looking at that mortality rate, I'm like, are we just not any better at this? That's the number of people per 100,000 who die of cancer. So
we are.
It could be a coincidence, but it bothers me that it's the same number.
But it's over many years that we have insurance companies here in the United States paying for not treatment, but detection, right? So we have more testing, we have better testing. People are actually going to the doctor more, especially since the America Cares Act. People have insurance, people are going, they're getting tested. You have cancer, right? As opposed to just dying and not knowing why, right? So now we have better record keeping.
We have better record keeping. And that's actually what's disturbing me, because that's saying, even though we've doubled our detection rate, our mortality rate hasn't really changed. The only reason it's down fiber. 50%, which is sounds like we're winning, is because we've doubled the rate of the number of people who we've diagnosed with cancer.
Yeah.
Well, the actual number per 100,000 hasn't moved. That's frightening. That's terrifying. Well, that's not good news at all.
I thought you said it was down by 50%. That's good.
But that's because the diagnosis
percentages
rate is up, doubled.
Numbers. I know. Yeah.
Anyway, yeah.
Yeah.
So but it may just be a
That's
giant
good
coincidence.
news.
Maybe it's not.
I don't think so.
How come none of my good news stories ever end in good news? It started great, cancer's down 50%.
Because you pick them for that
Yeah, I
reason.
was gonna say, Justin, you have all the power here.
No, the problem is I never read the dang story to the end. I'm like, oh, this looks good. I'll put it in there. It's good news.
So
And then
you're
I read
learning about it with all of
Lies.
live on
us.
the
I
air.
see. Yeah.
Lies, lies, and damn lies. Oh my gosh. Well, I got a lizard.
Yeah, let's hear about this lizard
Let's hear
snake.
more
Give
about
me
this
it.
Yeah. Okay,
legless
so
lizard.
well, it has legs. It's not a snake.
So then it's a lizard.
It's a lizard.
Lizard,
Wait a second.
but maybe it is
This
like
whole
an early
talk about
ancestor.
lies, damn lies, and more. Whatever.
I mean, it would not be as exciting for me to say, I've got a story about a fossil squamate.
Yeah, it would be exciting to me.
Okay, back up for those of us who don't know what a squamate
All right.
is.
Squamates, lizards and snakes. Lots of them around the world. They maybe originated about 190 million years ago, much younger than those fungi you were talking about. Researchers have found a new fossil, but it's old, in Scotland, and they have called it Brugnathar elgolensis because it was found in Scotland, and so that's what you do. They have taken Brugnathar and compared it based on its morphology and other aspects that they have been able to measure with in-group and out-group phylogenetic comparison. And they're like, oh, okay, it's definitely a lizard, maybe early snake. It's very similar. It's got a jaw like a snake with big fangs, like big. What are the big fat snakes?
Like
Anyway,
a viper or
not
a
a viper, like
or
no, the big ones that are round
Boa
and like a
constrictor?
boa, kind of
like the anaconda. Sure.
Anaconda. Yes,
Yeah.
got an anaconda because he's got.
So
anyway.
boas and anacondas are constrictors, so they have they're tinier rear fangs. They don't have the big venom injecting fangs in the front.
Yes.
Anyway.
Yeah. So they had a big jaw and these kinds of large snake-like fangs from an older snakiness, but it also had little tiny legs that were very lizardy and gecko-ish. So it was kind of this. It had like old lizard traits, reptile traits, but also snaky traits. And so they were like, who are you? And so they gave it a new group. And so Brugnathar elgolensis has been added to a new group called the Parvoraptors. And these Parvoraptorids are called raptorids because they are thought to have hunted prey similar to raptors, like the dinosaur species, velociraptors. But the Parvoraptors puts them within this squamate group, apparently, because of the way the words work. So anyway, the clock for this particular find takes us back to the Middle Jurassic, 167 million years ago in Scotland. And it's a really great, almost complete fossil find. And because of the way they were able to see all of these traits, they're like, this isn't here in more modern groups. This is from older groups. And they've been able to look at these snake-like features, the teeth and jaws, and then also with the gecko, leggy features. And there are comparisons in the phylogeny that suggest that it could be more on the snake side, or in a different phylogenetic analysis said, no, it's more on the gecko side. And so it's a really interesting thing because it's still not answering the question of where snakes came from, but because of the dating and the particular traits that they are seeing, they are saying that this particular group of these parvoraptorids, are taking us back to a very, very old age within the Jurassic. Co-occurring with dinosaurs, raptors, and others. So these squamates, the early where the snakes and the lizards come from. Anyway, there's traits. They have this is a great fossil find. They're really excited about it because it potentially takes the question of the origin of snakes versus reptiles or lizards, and it's getting at is this convergent evolution, or is it actually origination? And so that is the question.
So sorry, where on this diagram is this new guy?
So for the phylogeny here, the stars, we've got a couple of stars, and depending on the phylogenetic comparison that they did, they put this dude in different groups. So either
that's extremely confusing.
Yeah, it's super confusing. They're like, oh, we compared it with this one data set and we compared it with this other data set, and depending on who's included and what data's in there, this dude could be kind of anywhere. So it's a mystery wrapped.
So this is So this is interesting because I have learned just from kind of learning more and more about the evolutionary history of reptiles in particular, that reptiles are a junk group. There's no such thing as a reptile. It's polyphyletic. Right. It's a mess. But evolutionary time has shown us that a lizard-looking thing is a successful body plan. A lizard-looking thing keeps happening. Right. It's something that evolution returns to over and over again. And so it's understandable that, when people were first making their buckets, they were like, you're a reptile and you're a reptile and you're a reptile and you're a reptile, right? Yeah. But of course, like, crocodiles are more closely related to birds than they are to lizards. And then there's the tuatara, which is what I really wanted to see on the phylogeny you just showed. Rhynchocephalia is called out on there. So that's the tuatara, which is not a lizard and is the most lizard-looking non-lizard you've ever seen. But based on looking at their skull and the arches in their skull, that they are completely unrelated, or at least they're not a modern day lizard. They came about in the Triassic. And so this is a very similar situation where they're like, you don't quite fit into any of these categories, but you look more like this.
However,
and this is not genetic. This is all morphological, and
that's limiting.
And so it's limited.
However, this is older than snakes.
Well, it's going back. So the question is the stem group, right?
Go back to that horrible graphic. Go back to the horrible graphic.
At what point in time did snakes and lizards splinter, right? At what point in time was there that differentiation? Yeah. And there's still a massive question about this.
Hang on, no. There's a point in time when snakes have limbs. There's a point in time when snakes had, walking around on 4 little feet. We don't know what they look like. Maybe they were gecko feet. They were likely not hooved, in any case, right? And so if we're looking at this, it's around. It looks like it's pinpointing itself before the advent of snakes, time-wise.
There's three stars, Justin. There's 3 different places that they think it might have landed.
Right. And these all look like they're somewhat before the advent of the serpentines on this list, or our known history of this.
One is a branch off of serpentes.
Yeah.
Right. One would be very closely related and and kept the legs, right? And then there's others that are building off of other forms, because again, this lizard form, like you're saying, is so conserved that there's lots of versions that kind of look like somewhere in between. But in any case, what we could be looking at is a form of what a snake might have looked like before they lost their limbs. And I think that's a very fair way of analysis of looking at this creature. It's not necessarily the snake predecessor, but very much a picture of what the snake probably looked like before losing their limbs.
I love the end of the abstract says, these findings indicate high levels of homoplazy and experimentation during the initial radiation of squamates and highlight the potential importance of convergent morphological transformations during deep evolutionary divergences. So they covered all their bases.
That's what
there.
I said.
They said, we don't know. Yeah, that's exactly what you said. I know.
Yeah, but all of
said it, but not as smart.
All of their stars are showing something that is in the ancestor pool of snakes.
And hey, they're all from Scotland. No, I don't know. I'm going to just say very, very quickly that I'm done with that story. Everybody, thank you for joining us tonight and enjoying this adventure through science land and the internet. And I hope that you are just having fun with us as we are having fun here with you. If you enjoy TWIS, please share with someone today. Make sure you head to twis.org. Help people subscribe to our podcast, to our live streams, to our channels, YouTube, Twitch, Facebook. And Patreon is where you can support us in an ongoing fashion in a way that, helps us not be able to fix the internet issues that plagued us this evening, but, hopefully buy us new microphones when we need them and keep our cables working well. So glad that you're all here. Thank you for your support. All right, coming on back. It is time right now for that part of the show. What? That part of the show. We call it Blair's Animal Corner.
With Blair.
Yeah, and I'm going to hit the music button again soon, but I realized that I didn't set everything up the same way that I did from the last one. So here it goes.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, goodness. I think I'm going to start with some crabs.
Lady crabs.
So
Blair's got crabs, everybody.
Yeah.
Crabs.
This is a study from the University of Portsmouth Institute of Marine Science, and it is looking at crabs and their sensitivity to electromagnetic fields. Now, this is not some like crystals and incense situation. Underwater power cables actually emit electromagnetic fields, and there's generally an expectation that it doesn't do much. They're pretty low amounts of energy that are releasing from these power cables, but millions of crabs migrate along coastlines every year to reproduce. And so they are walking directly over, through, and around these electromagnetic fields. Now, just, as a Californian, and also as someone that, used to work at an aquarium on Pier 39, which is a crabbing, very heavily influenced kind of crabbing zone, I knew right away, like, oh, well, that's interesting because the take of crabs is very carefully monitored because crab mating seasons and the time when they lay their eggs is very specific. And so, in fact, you only can take male crabs. You are not allowed to take female crabs. If you have female crabs in your crab traps, you put them back. And there are specific seasons because you don't want to take crabs during mating season. You don't want to accidentally catch a female that is egg bound. And so there's lots of protections to make sure that the crab fisheries do not, collapse. Lots of people depend on crabs, and also a lot of animals, of course. Not to mention the fact that crabs are detritivores, and so they're like the janitors of the ocean also. So pretty gosh darn important ecologically. Well, what researchers found was that crabs do have a sensitivity to electromagnetic fields being emitted by these power cables, but specifically only female crabs have sensitivity.
Why?
Yeah, so they grabbed some crabs, popped them in the lab, 120 juvenile common shore crabs, and they exposed them to electromagnetic fields, covering kind of the intensities that you would find in underwater power cables. They started very, very low, and then they kind of ratcheted up to what you'd find in the high end. They showed significant behavioral differences between male and female responses. And of course, my assumption would be, oh, they hated it, right? No, female crabs were attracted to it. They were twice as likely to stay near areas with electromagnetic fields compared to those without them. So this is an issue not of hurting them, not of repelling them, but of attracting them so heavily that they don't move and disperse.
So they stick around the cable in the electrical field and don't go being excited by actual crabs.
Yeah. Males, meanwhile, not affected, no consistent spatial preference at all across any strength of electromagnetic field. They don't care. Yeah, and so they did, like I said, expose them to a wide variety of kind of levels of electromagnetic field, and they did find a statistical significant change in behavior for females, even at the lowest amount and also at the highest amount. So it was just across the whole span, they found responses from these female crabs. They did 10-minute trials, they tracked their behavior, they looked at zone preference, mobility, all this kind of stuff, and they used some tracking software. So they found that the adult female crabs, their movement was reduced by more than a third, 38%. And this was at moderate field strengths, which is about 1,000 micro taus? Electric units.
Teslas?
No. But they also found even noticeable effects at super low. And so I was just like, what kind of electromagnetic fields impact people? And so it depends what the source of the field is. It depends what the type of emitting is. But generally with electromagnetic fields, the main concern with human tissue is heating it. And for there to be any impact, it actually has to be like at least 10 factors higher than any of these highest ones that they were testing on the crabs. So
that makes sense. That's for like heating your tissue. That's like actually,
yes,
but not
for damage wise, it's not.
But not your sensitive electrochemical nervous system.
Right.
Responding.
So I don't want to. I know this was a study in crabs, but have you guys noticed there's a lot more electricity in big cities? And there's a lot more people.
Oh my gosh!
I'm
going to ask though. Like we do know that sharks and many fishes have lateral line systems. That the electromagnetic sensory system is very important. And it's interesting that like sharks that are predators, what other predator species are tuned in to that electromagnetic sense? And then why just the females? Have we ever looked to see if males emit anything in that electromagnetic spectrum?
Yeah. I mean, so it's there were a few things that kind of struck me about this study. So one is it's a very good reminder that when we put things in the ocean, we like to assume it's just like, don't worry about it. It's over there. It's underwater. You can't see it. It's all good. So there's that. There's definitely, it's, whether it's intentional or not, there's a little bit of that. But also underwater cables. So they're currently less than 0.1% of the ocean floor.
But they're all covered in female crabs.
But it's important to place them in the right place. If you are placing them along movement pathways of crabs, is that going to be a problem? Are you placing them in a shark nursery? Right? Where are they getting placed? Because often these underwater cables, more and more of them are going in as part of offshore wind farms. And so specifically, this is part of the effort to move towards renewable energy, which is, of course, important. But when we do that, we need to make sure that we're also paying attention to, how we're impacting. But the other piece of this that I think is a good reminder is the fact that the males and females had such drastically different responses. So it is another reminder that you cannot study literally anything on any animal subject without testing both males and females, because you have no idea. No one could have predicted that male and female crabs care more or less about electromagnetic fields. Like, that is so unexpected and such an excellent reminder that you have to test both. And especially in this case with crabs, you don't want to pull the females from the wild because of fishery rules. So, like, do you get your extra permits? Do you pull females? Or do you go like, oh, I don't want to make that ecological impact, I'm only going to pull males for my study? Well, then you think that electromagnetic fields don't matter. So it's still really important to do that work.
I immediately have like 3 questions.
Yes.
One is in the chat room. How do you
tell them apart? Yes.
Yeah,
so
male
it is.
Female crab.
So if anyone wants to Google image search male versus female crab, I'm used to looking at it in Dungeness crabs, but this is true in other crabs too. Think about a crab is a squished lobster, right? So a lobster is a tail. The crab has a tail also, but it is bent under and flush with the body. If you pull the tail off, it looks like it has a perfectly fit kind of like indentation that it lives in, right? Mm-hmm. And so the tail is a completely different shape for males and females. I don't remember. I think the female is super wide. It looks like almost like a circle, and then the male it looks like more like a pointy triangle. I think that's the difference. The male is the pointy and the female is wide because all the eggs are hidden under the tail.
Yeah. Okay. So you just look at the tail itself.
It's very obvious once you know what you're looking for. Yeah.
Okay. Next question. The next thing that occurred to me is, could this be used as just the new habitat? Is it perfectly acceptable for them to be in this location? Will the males go and find them? Can this just be like, this is now where crabs live? And that's fine.
So you want dispersal. You don't want all the crabs in one place.
You don't want to put all your crabs in one crab basket. Okay.
Yes, that's
And correct. Then the next one I was thinking is, does this have anything to do with crab preference? Or is it a crab preference? Like the crabs are detecting it, right? The female crabs are detecting something in the electrical. But why? Mm-hmm. It's probably not a preference for an electrical field. Right. It's probably a signal for something else. Something that they would maybe a nutritional factor, something that they would eat that's normally itself attracted to or generating.
Yeah. So Kiki brought up a good point about that. With sharks, for example, their lateral line detects electrical signals because of like heartbeat, basically. So they're sensing the chemical processes of life. So like they're sensing the electrical current of life. Yeah. So that could be it. Absolutely. It could be that crabs are like, ooh, little fish I can eat. Krill, right? Like totally, that's possible. But they are detritivores. They usually like poop and dead stuff. So I don't know. I'm not sure. I think that's a really good, rich future study is figuring out what it is about the electrical signal. Is it just something that's inadvertent that's triggering a chemical pathway in their body?
Like I would, my point would be to go look at existing crab fields and check for like where they tend to congregate, and then just see if there is some sort of background electrical field being generated from something in there that they're already being attracted to, that we just maybe it's weaker, maybe it moves, and if it moves, is it moving with currents? Is it Kiki, we can't hear you, so
sorry, but this is weaker and it's getting in the way, right? So it's it's stopping it is attracting and disrupting the natural behaviors of these crabs, and that's bad. So Blair said Let's be smart.
Mm.
Like with the dodo.
Yeah, speaking of being smart, why are we bringing back the dodo? We're not actually, but Colossal was in the news again, and I just wanted to bring it up to kind of tell everybody what's going on, because they were in the news again, and I wanted to talk about what actually happened. So, the headline was, Colossal makes huge leap in bringing the dodo back from extinction. What they did was, they cleared an early hurdle by growing primordial germ cells, which are the precursors to eggs and sperm, from the rock dove, aka pigeons. So that's it. They created pre-gametes. They did it.
But rock dove pre-gametes, not dodo. It's pigeon.
Yes, because
they're related, and
right? They want to have the primordial germ cells stable enough to be able to gene edit them at that stage, because with mammal reproduction you can edit like DNA in an egg. Okay. But you can't do that with birds.
Because of the shell and all the things.
Well, yeah, and because the egg is actually developed much later. Like the fetus starts developing before there's an egg, basically. Because the egg is like it's coinciding. The egg is developing as the fetus develops. And so because of that, they can't use the same gene editing techniques of how they would try to do it, like with the southern white rhino and the northern white rhino egg. Like they can't do it exactly the same, right?
Or like they reported on the dire wolf, right?
Exactly. So they did the dire wolf thing. Yeah.
Wait, hang on.
Not real, but yeah.
So this actually could be a major. Are you saying this is a major breakthrough for bird conservation?
Pigeons.
It's a pre-breakthrough. Because all they did was pigeons. All they did was make pre-eggs out of pigeon. I would argue the big jump in bird conservation would be editing that pre-egg. But they haven't done that yet. That's actually the thing that's going to get you anything. Right now, they're just like, we almost made an egg. Is basically all they did. Which, that's not even their plan. They're not going to do the rock dove, the common pigeon. They actually have to do the Nicobar pigeon in Texas, which they have established a breeding colony of for this activity. That's the one that they're going to have to do, because they are saying that is like the closest living relative to the dodo that they have, is this Nicobar pigeon in Texas. So they have their germ cells for editing. Then they're going to be able to edit what they have in the Nicobar pigeon germ cells, and then put them into chicken embryos. And then the chickens would grow up with pigeon making cells in their ovaries or testes. They would be the surrogates. They would lay an egg with a dodo in it.
With an organism that is an approximation of the genetics, the genetic representation of a dodo.
Correct. So it's actually, it's 3 different birds. Yeah. But one of them is a guess, basically, because we don't have a dodo genome. So they're basically just like, they're going to fiddle with the chickens and the pigeons until they get something that looks like a dodo, is what they're trying to do.
Wait, why don't we have a dodo genome?
We do, but that's why they're gonna. But they have to mess with the pigeon genes to be like, they have to get it going so they can edit the pigeon and chicken stuff to be able to be like, we're putting in dodo genes. And
so we have a pigeon genome, but it's not. You can't just inject that into. You have to make it functional with the pigeon genome. So you can't just go, I'm going to put this in here. Exactly. So you're going to have your
a new generation, and then you have to do it again to add more dodo gene in. So it's going to be a fast track of genetic evolution in a lab.
But to Justin's point, Colossal's chief science officer says, by developing these protocols, we're establishing crucial biobanking capabilities and opening new possibilities for genetic rescue of endangered species. So, as we've said before, this like PR nightmare is really just fueling their ability to do just groundbreaking conservation efforts later. All that to say, they say that they have a place to put these dodos, which I think is bonkers. They're going to put them on an island country off the coast of East Africa where birds were endemic before they went extinct. So they've like identified habitat for the dodo already, which I think is insane. But I just wanted to bring to daylight that once again,
I mean,
this is
look,
like
that's to be fair, you like if they hadn't identified at least a place where they would plan on putting them, you'd be like, and they don't even know where they're going to put them. Like,
no, let's be real. They are not going to make a functional. They're not going to make a functional population of dodo. That is not going to happen. They're going to have their 20 dodo that they have in zoos and they parade out at the bioscience conference on a leash. Like that is what's going to happen with these animals. They are not going to reassimilate into the wild. That's not how, let's be real. That is not what is going to happen here.
Okay. But the technology they develop could return a variety of pigeon, perhaps, back to the wild.
Sure.
After
absolutely.
It became accidentally
I am not
extinct.
saying it's not like sound science, but I think, as is always the case with these colossal articles.
Yeah.
I want to, put some daylight on this and say they are not bringing the dodo back from extinction. Even if they made something that looked like a dodo, it wouldn't be the dodo that they were bringing back from extinction, just like it wasn't a dire wolf that they brought back a couple months ago. It is not. It is an approximation.
That one did hurt my heart a little bit.
And so they're not doing that. They're also, like I said, they're not going to bring these guys back to the wild. They're not. This is all proof of concept that's like sexy and exciting for people to talk about in the news. Cause like, the dodo. I would love to pet a dodo. Oh my God. I'd love to feed a dodo a worm. Like it would bring so much joy to my heart. But it is also important that we remain grounded and recognize what's actually happening.
Sensationalism, PR in the service of science.
And we'll be damned if we give them any publicity here.
Yeah, they're gonna have.
Even though I've said their name 20 times.
Many patents. Oh my gosh.
Yeah.
Wow.
Hey, one bright spot.
I'm gonna tell a story.
By mixing it with the chickens, I had always heard that dodos tasted terrible. So maybe
now they'll taste like chicken.
They must have tasted great.
No, my understanding was it was just a matter of they were easy to catch. Because they weren't afraid of humans, but that they actually it's just sailors stuck on an island,
Tough and greasy.
Yeah,
sure.
Apparently,
but you better believe Charles Darwin ate one. He ate.
Now
everything.
they will taste like chicken. Justin, you want to talk about something techie?
Yeah, I brought a tech story today, which is I've never heard of this. So everybody needs to Google the G1 humanoid robots.
Whoa. What?
This is that human okay, I'll just read the story because I'm not hip to the fact that we've got humanoid robots running around, but it says,
oh no, it's this one. I hate this one. All right, I'm sorry. I just looked at it.
Researchers have it looks like a Cylon. Like the ones without the skin
except black. Right? Like the black, yeah. When they were silver and black later.
Yeah.
Okay, yes. Go ahead. Sorry.
Researchers have uncovered serious security flaws with the Unitree G1 humanoid robot. Something that's already being used apparently in laboratories and in some police departments.
What?
That's what this story is saying. I believe every word I hear, but I don't. Oh my gosh. They discovered that G1 can be used for covert surveillance and could potentially launch a full-scale cyber attack on networks. So this is all
all within China, right?
Is it Sounds like the stuff of science fiction nightmares, robots that are secretly spying on you and could be controlled by remote hackers. However, the concern is real as these types of robots are becoming increasingly common in homes, businesses, critical infrastructure, and public spaces. So this is the article is by Paul Arnold over at phys.org. It says, in a new study, cybersecurity experts from Alias Robotics describe how they performed a digital audit on G1, reverse engineering its internal software and eavesdropping on its internal communications to identify critical weaknesses. One of the most serious flaws was its Bluetooth low energy setup for connecting to Wi-Fi, something that's apparently common to many consumer robots. So they found that the encryption protecting this process was incredibly weak and easily broken, as it relies on a single secret digital key hidden inside every Unitree robot. And simply encrypting the word Unitree with a hard-coded key was enough to bypass security and gain control of the robot's entire system, which means hackers could easily take it over, inject some malicious commands to crash it or make it attack other devices. Equally concerning was that G1 acts as a Trojan horse, secretly and continuously sending data to servers in China every 5 minutes without any sort of acknowledgement to the user that this is going on. Team showed that G1's onboard computer could be repurposed for offensive operations. Additionally, robot's custom encryption method to protect its internal configuration files is fundamentally flawed because, again, simple static key that's the same on every single robot. So if you figure out how to unlock one, you got them all.
Oh my god. So this is the other thing. I just looked these up. They are not as expensive as I thought they would be.
No? You want one now?
No, absolutely not. Never in a million years. But no, I mean more that just like if they're so easy to hack, but they're relatively cheap. They're like cheaper than a car. Like just I think that compounds the issue a little bit. That, you know, if they have their way, there'd probably be quite a few of these running around, literally.
It seems though like this is a basic technology issue, right? That networked devices, we have the back doors, we have like issues related to the DJI drones across the country. Like they just reassessed and said, nope, they got back doors. You're probably giving data to China, so people still aren't allowed to use their DJI drone technology. But these kinds of devices, if they're being put all over the place in sometimes, you know, high security or in, you know, areas where
can't trust a human with the job.
information.
Oh, we'll get a robot. It can never betray us.
It's crazy. Like it would take so much work for a human to like record something or like copy something and share it.
Send it to their secret contact in China.
But meanwhile, you have the robot. It's just like, oops. Just it's
every
open.
5 minutes. Every 5 minutes. That's wild.
Oh.
Wow. And you know, this is unrelated, but I'm just gonna quickly throw out there. Can we stop making robots look like humans? They don't need to be bipedal. They don't need to look like that. They don't need to have a head. Can we stop? Can we, and especially until we figure out how to not hack them, can we stop?
Like headless robots. Okay, everybody.
I want something with like a tank tread situation, like more like Rosie from the Jetsons. Can we do that instead? Could we just move towards just like the arm on a tank tread? It's not make it look like a human. That would be my preference.
I don't mind them making it look like a human as long as they make it out of cheap plastic. Right? If the future robots are made out of some kind of cheap lightweight plastic, so if they do turn into murder bots, you know, and they try to attack it, they got like
just
no
give them a
limb
nice
strength,
like little swipe, trip them because their
just
bipedal design is really dumb. Actually,
cheap beach chairs from, you know, the variety store.
Exactly. Don't give me the tank. Give me some cheap plastic robots that are easy to push around, and I think I'm less scared of them being murder bots. But now that I know that they're betraying us, our every secret, and the other thing is like that idea of the connectivity in today, like where things are like connected to your central computer, you can turn your lights and stereo up and down, and everything's all connected. And you put this robot,
smart homes,
in that smart home network, and then suddenly it's compromisable and can turn everybody's lights off at once, or disconnect your refrigerator, or order stuff on Amazon for you. Like, it becomes a problem.
The problems you just listed were ridiculous. Oh no! Bad networked robot turned off my lights.
No, everybody's lights. Everybody who's connected to, like, across the nation. Like, you got to understand, this is like in every. It's like a really common Doctor Who theme, where like something that becomes ubiquitous within society that everybody's like, oh, this is a normal thing, and then you find out there's like some sinister alien plot behind all of it all along, and then turns on everybody.
And I totally agree.
Step one.
There is the TV
Step
show,
two, murder bot mode.
It is one of the best book series. I love this book series so much. Murderbot Diaries, highly recommend if you have not read them. Very fun if you like murder bots. Yeah, it's great. It's good. Imagine blood. It's fantastic. I have two stories that I will get to over a very long time because it's early still. No. Think of this show like a super centenarian right now. The length of the show is like the lifespan of the world's oldest people. What is it that defines the longevity of those individuals, and what is different about those individuals from the people who just live normal lifespans or shorter lifespans? So researchers just published in Cell Reports Medicine their analysis of one individual compared against a whole bunch of other people. So they did a complete multi-omic blueprint of a super centenarian, female aged 117 years old and 168 days. Caucasian woman born March 4th, 1907 in San Francisco from Spanish parents and settled in Spain since she was 8. She had a lot of great life experiences. She lived in Catalonia. There's another place, M116, another long-lived individual. Life expectancy in Catalonia for women is 86 years. So that's exceeded by more than 30 years. So what's going on there? And so these researchers decided to look at everything. All of the genes, all of the proteins, all of the transcriptome, so RNA, DNA, proteins, microbes, everything. They looked at it all. And what they found is that the majority of the features of this individual were those of a younger person. Immune response, microbiome, epigenetic clock, the inflammation, cardio, metabolics, all these things were similar to someone who had not really gotten into the downward slide of aging yet. Yeah, hi, what do you want?
Well, I don't mean to interrupt the beginning here, but when you say younger person, I did mean to. I raised my hand and I started talking.
You did. No.
When they say younger person, I'm wondering if they mean an 86-year-old.
No.
Right? Or somebody who, like, younger than 117 leaves, like, everybody.
Right, it does. Absolutely. Okay, so the comparison that they did, they they were really looking. They compared this one individual to like massive database of multi-omics of individuals, and so the comparison that they found is the chromosomal clock. They found that the telomeres, the short telomeres, they had a smaller number of short telomeres. They did find, and they say, actually, no, I said that wrong, and I'm sorry, I'll correct myself right now. We talk about telomeres being the ends of the DNA, the protective bits that get worn down as cells divide and that protect our DNA. This individual had a huge erosion of telomere sequences. So what's going on there? So is the telomere attrition more of a chromosomal clock rather than a predictor of the diseases or the aging that's going to come? Wow. So it's a different way of looking at telomeres.
That would be wild if we kind of completely shifted our thinking on telomeres because of 117
they're involved,
year-olds.
But let's reframe it, right?
I'm glad that that's there because it's evidence that this person is of the age. I'm always suspicious of
they are old.
Well, it's because like they have to have, started life in a time with really poor record keeping. And
yeah, and you reported on the blue zones and how those were all kind of poor record keeping. Yeah.
Well, and then like all the 20-something-year-olds in China come from a region that only started tracking births in like the 1950s. So like they had like makety up birth certificate times or ages, right? They were inventing their own birthdays once people started coming around and creating IDs. Like they didn't even have IDs at that point. So then I'm like, San Francisco, Great Fire, records can't have been there. Oh, maybe the family fled to Spain after a murder and it was like, oh, our name is this and this is our child so-and-so. And they're like, oh yeah, she's really a child. She just looks, whatever the thing is, right? Like, no, I'm always suspicious of age. And now hearing this highly deteriorated telomeres that's tracking with a 117-year-old makes more sense to me than them just having younger features. Because then I'm like, they're just not old.
Right. So they are older, right? They are older, but they've got aspects that are similar to younger individuals. And you asked about the age, and the younger individuals are around 25 years old.
Wow. That's incredible.
Yeah. So these individuals who are living 110 plus, right? These individuals really, they are younger forever. They're not aging the same way that everyone else does. So this woman, she did still have age-associated B cells. There were immune cells that were related to aging and being older. There were issues related to blood volume, hematopoiesis. So the ability of and how your blood volume is being turned over. So there's stuff that was getting older, right? And it was not super young anymore. And there were mutations, which is what you expect. As you get older, as you live longer, you accumulate more mutations. So there should have been more mutations found in certain places in this analysis, and they found them. But it's really very interesting. There's the good living aspect of it. I mean, Catalonia, probably not a terrible place to live when you're, probably was great. Mediterranean diet. I don't know. She lived through a couple of world wars. She lived through flus. She lived through massive pandemics, global flu pandemics. She lived through a lot of things. And still was younger than me.
Tell the tale.
No.
So the lack of, or the low grade inflammation, the lack of inflammation is really interesting. My guess, I'm going to put out a guess that this is going to come down almost 100% to mitochondria.
Possibly, right? Or at least I mean, the genetic profile or the immune system that allows the mitochondria to stay healthy, right?
Before that, it's going to be before that. It's going to come down to the ATP and the what is it? It's okay. So it's
but also getting rid of trash.
The nucleotides. It's within the mitochondria build up. The precursors for RNA tend to build up over time, and this is where you start to see senescence of cells and the what is it called, that signaling cascade that sets off the alerts that starts inflammation. That whole process begins in mitochondria and has a lot to do with whether or not it has accumulated too much precursor, because mitochondria doesn't normally have a way of getting rid of this. Now, her mitochondria might be able to balance itself and expel these precursor RNAs so that they don't become burdened, so that balance stays good. And so those mitochondria, we got to study and figure out how she's getting rid of it, because that could be the key to unlocking the fountain of youth.
But you're guessing at this when this is an entire systemic analysis, and
it looks like it.
The mitochondria are very, very, very important. I mean, really, truly.
But I'm gonna predict that that's where this is gonna end up, because this is not part of the study, but there have been these studies that have looked into the building block nucleotide buildup in mitochondria's effects on inflammation and aging of cells. Like the main things that we see as the aging of cells begins with and maybe even ends with mitochondrial health.
But if the
mitochondria,
immune system and the garbage, the waste system is working fine, then the mitochondria can stay healthy. I mean, that's one of the aspects.
But they still age. I mean, that's where aging happens. That's where the senescent cells happening. It's all mitochondrial health that affects cell health, that affects tissue health, that it expands from there.
Yeah, I mean, she was old and she died.
Because I needed to study her mitochondria.
Aging happened. This wasn't a mitochondria study. This was just looking at this person, and I like your hypothesis.
That's why I'm like, how is that not? So this is after the fact that she's gone, which is unfortunate. They need to find those super agers now and do just a complete mitochondria study on them to see if it is. Woo.
I think that is that could be very interesting. I was pointing up to where Blair is in my thing, because look at her face. She's so tired. I'm trying to finish.
I'm fading fast.
I know. I have one last story, and that's it. That's it, everybody. One more story. Maybe if you want to have good memory, don't eat junk food. That's it.
What'd you say?
No. Did you forget already? Oh my gosh. What'd you have for dinner?
Cookies?
Yeah, researchers at UNC School of Medicine have just published a study in mice, not in people, but in mice, because they're looking at mouse metabolism. But oh my gosh, when they gave the mice high-fat diets, that were supposed to be like our highly processed sugar fat diets of the West, they found that it led to a change in the way that the neurons used glucose, and they became less likely to do the job that they were supposed to do. And so neurons in the brain, the interneurons in the hippocampus, which is part of the memory hub of the brain, they got weirdly active, but they were not modulated in the way that they should be by the amounts of glucose that were present. And so what they are suggesting is that there are very sensitive memory circuits. They're really sensitive to what we eat, and that this is not related to how much you weigh or whether or not you have a metabolic disorder like diabetes. This is specifically just brain glucose levels, fat in the diet, and the response of these neurons to what they are saying is, mouse junk food. They did find, however, even though the neurons got all messed up and made it so that basically short-term memory and all that stuff is not going to be great after you eat junk food. Hey, college students, like cramming for your finals, junk food during that period, maybe, eat a nice meal, go to sleep. Also, intermittent fasting, they found reversed the effect of the junk food. So the junk food messes things up, but when you go, let's start over with some intermittent fasting, and maybe there's other stuff you can fix it. If you eat junk food, all is not lost. Your memory will not be completely gone. But that fits
also.
I hope you like your cookies.
Also fits an energy use strategy, meaning we have a lot of energy now. Let's not waste any time remembering what's going on. Let's go hunt. Let's go fish. Let's go do whatever a mouse does with its time off. Let's go get more resources. And now that we're fasting, now we can think back on life and ruminate about things for a minute.
But if you think of the way that animals eat, and then normally you don't have junk food, right? Like maybe you are a shark, a great white shark, who gets to eat the seal candy bar, right? But with all the high fat and everything in the blubber. But more often than not, what you're dealing with is an evolutionary system that is set up to rest and digest after you've taken on a very high calorie fat meal, not keep going and act like that was lunch. 15 minutes standing up or walking on your way from place to place. So the way that we function is not the way that evolution or adaptation led us to where we are. Anyway, junk food.
Can you write me a note
Thanks,
for an extra long lunch break at my work?
Yeah.
Blair needs to process her food or her memory will be bad.
Yeah, if you want her to remember anything in the afternoon, you really need to make sure she has time to eat good food and
a 2-hour lunch.
Yes. Lunch, exactly. Anyway, might just be telomeres and mitochondria. I don't know. I hope everyone will remember this show forever. I hope you have all of the stories and the conversations that we have been having just in your brain, because this isn't junk food. This show is nutrition. Are we done? Ready to go?
Let's do it. Let's close it out.
That's what I was gonna do. Justin looked like he was gonna fight it for a second. I'm like, what is
no, I'm ready. I gotta be at work in a couple of hours.
I know. Thank you for listening, everybody. Thank you for being here. I'm so glad that you joined us for this extra long show that we were going to finish at 10 o'clock. Shout outs. Fada, thank you so much for your help with show notes, social media, everything that you do. Identity 4, thank you for recording the show. I know tonight's going to be a little bit weird. Gord, Arnlor, everyone who's in the chat rooms, thank you for keeping it wonderful places to be. Thank you for enjoying chatting and being part of it all. Rachel, thank you for editing the show and assisting in the way that you do. Thank you to our Patreon sponsors. Can never forget them. Hey, everybody. Thank you to Aaron Anathema, Adam Mishkan, Ali Viola, Ali Coffin, Andrew Swanson, Arthur Kepler, Artyom, Bob Calder, Bob Coles, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts, Dana Lewis, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Don Mundus, Donovan Styles, a.k.a. Don Stylo, E.O., Eden Mundell, Eric Knapp, Flying Out, Fredis104, G. Burton Lattimore, George Corris, Greg Briggs, Jack, Jason Olds, John Atwood, John McKee, John Ratnaswamy, Johnny Gridley, Ken Hayes, Ken Northcote, Kevin Parachan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessenflow, Noodles, Patrick Pecoraro, I hope you're doing okay, Paul D. Disney, Paul Ronovich, Philip Shane, Pierre Velazar, Pete I.G., Richard Badge, Robert Norland, Robert Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean Lares Lamb, Shubru, Stephen Alberon, Steve Leisman, aka Zima, Sue Doster, Teresa Smith, Tony Steele, Vagard Chefstad. Thank you, all of you, for supporting TWIS. We really cannot do this without you. Your support is essential. And if you want to support us on Patreon, head over to twis.org and click on that Patreon link on next week's show.
We will be back. Well, some of us. Wednesday, 8 p.m. Pacific Time, broadcasting live from our Twitch, YouTube, and Facebook channels.
Hey, do you want to listen to us as a podcast? Perhaps while you drive to pick up your fast food dinner that you probably shouldn't have for your brain? Just search for This Week in Science or where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here, today's show notes, links to stories will be available on our website, www.twis.org, where you can also sign up for Blair's newsletter.
We love your feedback. If there is a topic you would like us to cover or address, or a suggestion for an interview, please let us know in one of our social media accounts, or better yet, send us an email. Just be sure to put TWIS, TWIS, on the subject line, or your email will have no ties to a phylogenetic tree. It'll just be a bunch of dotted lines, and we'll never see it at all.
Yes, but we do look forward to discussing science with you again next week. And if you've learned anything from the show, please remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not.
good night, Blair.
Blair, it is amazing how fast you fall off the cliff.
Oh, yeah.
It is. I was watching it, and I was like, Justin, wrap it up.
and then I realized it, so I stopped talking, and then you went on and forgot.
I did. Then because I was excited about the thing, but
And
you
I'm
asked
like,
questions.
I'm
and then I
like,
had to
come
ask
on, can we
questions, and
just? I'm like, what are you doing, Kiki?
I swear, no. The very end,
Like-
when you're just calling it in, like reading off the screen with like
No,
the
and
flat
she was
tone
slowing down because she was trying to think of the clever thing at the end, and it was taking
so
up
good.
all of the processing speed. She's like, I need to come up with the clever thing at the end. I should have planned this ahead of time, but
Oh, no.
I'm
Never.
going to this slowly, hoping that I have time to
That
construct
was funny.
the clever thing at the end. Oh, and I'm now there at the clever part, pushing through to the end of this sentence with the clever thing.
Everybody, thank you so much for joining us tonight. It was a journey. We like made it. We went through it. We jumped from stream to stream. We didn't cross the streams. I might have in the background, which is probably what happened to everything. Say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin. Good night.
Good night, Kiki.
Good night, everyone. Before we go, I just want to say that I hope everyone who is a federal employee who is not able to work as long as the government is shut down is doing all right. I hope that we are able to continue to bring you good science to at least entertain you those long days of, not working. I hope you're not losing your jobs. I know there's a lot happening right now, and I just really am thinking about you. And Portland, Oregon, it's a total war zone. Don't come here. Oh my gosh, it's all on fire. I mean, it was until it rained, and then the rain put the fire out. It's okay.
I also, by the way, bigger topic, might not be back
don't say that. Like that's like not. Oh my gosh. Justin got a job. Then it may interfere with his schedule.
Like
And so
99% chance will be meaning that I won't be able to be here at this bat time at this bat channel anymore.
Right. So that means that we are going to have conversations moving forward about if we have to change the time, if what we need to do, if we're,
But I love doing this so late at night.
Yeah, no, late at night is fine. It just would have to be a completely
I'm
different
joking. Come on.
We could make it a little, I mean,
Ryan,
we can even make it
come on.
later,
He's so
honestly.
tired.
Fine.
Oh my gosh. I don't.
Bye,
Yeah,
everybody.
Hey, everybody. I just totally.
Maybe not on a school night.
Yeah, man.
Every night's a school night. I don't
I
know
know.
what time is. It doesn't matter
I
anymore.
haven't
But
been
You will
able
everybody,
all
to
be,
talk.
until
already-
they get sorted, you will all be missed.
no, you're gonna talk to me, and then I'll let everybody. And I know, but yeah, okay. You're missing everybody, but not yet. We're not making. I don't know what's happening yet. You have to let us know what's happening.
Goodbye forever. Oh, it was the best of times. It was
All right.
wonderful.
Okay.
Good night.
Okay.
We'll talk.
Everyone-
Yes, we'll talk. We'll sort it out.
I appreciate you so much. Thank you. Thank you. Good night. Thank you and good night. Everyone stay safe. Stay healthy. Don't get caught in a insurrection or anything. Do science, think sciencely, be good. Have
Eat cookies.
I don't know, stuff is good goes.
Eat cookies.
We are leaving now, so luck.